Hello, everyone. My name is Magda Markowska. I'm a PhD student at University of Warsaw, and I would like to present CONET, copy number event tree model of evolutionary tumor history for single-cell data. Deciphering the copy number evolution of tumors is very important for understanding of carcinogenesis because copy number alterations are ubiquitous across all cancer types, and they are most commonly associated with tumor hypermutability. They can also be important drivers of tumor evolution because they can delete tumor suppressors or amplify oncogenes. New technologies have enabled obtaining single-cell DNA sequencing data with relatively even coverage, but so far no methods have been published for inferring copy number evolution from this type of data. Here I present to you an overview of our model. First, we need to preprocess the data in the form of corrected counts and genomic beans for single cells. This we call the per bean data, and we transfer it into per breakpoint data, that is the corrected count absolute differences in genomic loci. We also need to establish the set of potential breakpoints. We use this data into our modeling procedure where we model structure of copy number event tree. Our tree is a non-binary tree with healthy cell as root, and then each node means one copy number event with start breakpoint and end breakpoint. We also model corrected counts differences distributions. The black distribution is the distribution where for loci where we do not expect breakpoints, and the expected value is equal here to 1. And the red distribution is mixed normal distribution with expected values 1 or more, because for the loci where we expect a breakpoint, the difference between corrected counts in adjacent bin should be 1 or more. After we obtain our final CONET, we use maximum likelihood to attach cells into the nodes of our tree, and it's important to notice that the cells can be attached to internal nodes. Also, you can observe that in our tree, the events can overlap and the breakpoint loci can repeat. When we have our final CONET with cells attached, we can use the per bin data in our copy number coding procedure. We have performed comprehensive tests on simulated data for many different scenarios, and you can see the results of breakpoint detection of our tests. False positive breakpoint detection rate is presented here, and it is 10% or less in all scenarios. Also, the false negative rate of breakpoint detection is very low, less than 10% in the most demanding scenario. And the last measure is symmetric distance, which means how many breakpoints we have inferred wrongly for an average cell. And you can see here that the results are 1 breakpoint wrongly inferred or even less. Here we present inferred CONET for xenograft breast cancer dataset. We had 260 cells to infer this tree. This is quite a complicated structure, but let me explain that first we always print the number of events collapsed into one node, and then we print the breast cancer genes that were concerned by events from this node, and the color signifies the number of cells attached. So our tree has quite a long trunk with 59 events that concern very important breast cancer genes. For example, they delete such tumor suppressors as CP53 or RB1. A second observation is that the tree branches, dividing cells into 2 distinct subclones, the one on the left with anti-RK3 unique event, and the bigger one on the right with those three breast cancer genes characterizing this subpopulation of cells. Here, you can see the illustration of our copy number calling procedure for the same data set. In the upper part, you can see the per bean data, so the corrected counts illustrated with different colors. And here the results. You can observe that we smooth the noise present in the biological data perfectly. And we also infer almost all copy number events, even very narrow ones, if of course they are present in enough number of cells. In summary, I would like to remind you that our model is the first Bayesian probabilistic approach for joint copy number evolution inference and copy number calling for single-cell DNA sequencing data. It is robust to errors in single-cell DNA sequencing data, thanks to joint modeling of per breakpoint and per bean readouts. For simulated data, our tests demonstrate excellent performance both in breakpoint detection and tree structure reconstruction, and we also infer plausible copy number events for true breast cancer dataset. I would like to acknowledge my co-authors, Tomasz Tonkała, Błażej Miasojedo, and especially my supervisor, Ewa Szczurek. Thank you for listening.